Hello, I'm Olivier Toumine, team leader at the Interdisciplinary Institute of Neuroscience in Bordeaux. In this paper just published in PNAS, we describe a computer model of Ampa receptor trafficking at synapses. Indeed, controlling the number of Ampa receptors at synapses is of crucial importance in synaptic transmission and plasticity. To better understand these processes, we built a unified quantitative model of Ampa receptor dynamics, which we systematically compare to experiments in a wide range of biological conditions. Schematically, there are two major mechanisms controlling Ampa receptor numbers at synapses. A vertical mechanism involving trafficking through endocytosis and exocytosis, and a second horizontal mechanism involving lateral diffusion of Ampa receptors in the plasma membrane, followed by trapping at the postsynaptic density, which we will abbreviate PSD. The basis of our theoretical work are single particle tracking experiments pioneered by the group of Daniel Choquet, allowing tracking of Ampa receptor diffusion in the neuronal plasma membrane. What you see here in red are the Brownian motion of quantum dots bound to Ampa receptors. Neurons express Homer GFP in green to label synapses. Note that Ampa receptors alternate between extrasynaptic and synaptic spaces. This experimental work allowed us to build the hypothesis and parameters of our model. We simulated by computer individual Ampa receptor trajectories. This 2-minute movie shows such a trajectory in red in a piece of membrane of 2 × 6 microns representing a dendritic area. There are 3 compartments. In black, the extrasynaptic space where Ampa receptors diffuse rapidly. In green, synapses where Ampa receptors diffuse more slowly, and in blue, the postsynaptic density where Ampa receptors are trapped by interactions with scaffold molecules. Ampa receptors can quickly switch from one synapse to the next. When Ampa receptors reach the borders of the region, they bounce onto it. Here we sum up 500 Ampa receptor trajectories in a region of 2 × 10 microns. Synapses are modeled as red squares. Note that Ampa receptors, starting from initial random positions, quickly accumulate at synapses. We have shown that Ampa receptors' enrichment at the synapse is proportional to the area of the synapse, in agreement with electron microscopy data, and also to the binding affinity between Ampa receptors and the scaffold molecules. Here is a zoom on a single PSD, where one can see better that the Ampa receptor traffic in and out of the PSD, explaining part of the fluctuations of Ampa receptor currents at synapses seen by electrophysiology. In the middle of the movie, the diffusion coefficient is set to a low value to mimic cross-linking with antibodies, which results in a stabilization of Ampa receptors at the PSD. We also simulated experiments of fluorescence recovery after photobleaching of recombinant Ampa receptor subunits. Here, the synapse in the middle is photobleached, and recovery of fluorescence occurs because Ampa receptors diffuse and exchange between neighboring synapses. We thus directly reconciled FRAP and single particle tracking experiments within the same framework. This allowed the estimation of 2 important parameters, the Ampa receptor scaffold kinetic rates, which bidirectionally control the steady-state Ampa receptor density at synapses. To reveal the impact of recycling processes, exocytic and endocytic events were introduced in the simulations. Here is an example where a single bolus of 50 Ampa receptors is delivered within the synapse in the center, very close to the PSD. Note the rapid accumulation of Ampa receptors at the PSD, which is stimulated. This is followed by a gradual loss of Ampa receptors, which exchange with neighboring synapses. Here, an endocytic zone is positioned close to the PSD. Ampa receptors are gradually removed specifically at this synapse. Based on these examples, the main prediction of our model is that synapses endowed with internal endocytic and exocytic recycling mechanisms are privileged over other synapses because they can quickly and efficiently regulate their Ampa receptor level in response to potentiation or depression stimuli. This model allows then for the first time to discriminate on spatial and temporal basis the relative contributions of diffusion, trapping, and recycling events in the regulation of synaptic strength. Thus, by combining experimental data and mathematical modeling, this study provides original quantitative information on the biophysical processes underlying Ampa receptor trafficking in synaptic plasticity. This general framework can be adapted to other membrane receptors in a wide range of biological situations. I would like to acknowledge my collaborators who contributed to the success of this work, and thank you for your attention.